Hello students, welcome back to your biology class. Today we will discuss about next part of the cell, that is cell wall. So let's start with the next topic, that is cell wall. Cell wall, as you all know, that it's a outermost membrane of the cell in some organisms, like in the case of plants, prokaryotes, fungus, etc. We will discuss about it. This is the outermost membrane of the cell and it works as a protective membrane also. So let's see what are the characteristics of the cell wall, or what are the important function of the cell wall. It is outer membrane. It is outer membrane. It is protective in nature. It protects the cell from the injury. इसका क्या काम होता है? It protect the cells from injury, and it protects from some external factor also, like gravitational force, etc. So its work as a protective. It provides rigidity to the cell. Itself is a rigid structure, so it's provide rigidity to the cell. You already know about the osmosis and plasmolysis. At the time of the plasmolysis, is protect the when water enter inside due to the osmosis. When water enter inside the cells, it's the size of the cells increase, so it's protect the cell by bursting. तो उस टाइम पे जो सेल की प्रोटेक्शन होती है बर्स्टिंग से, वो भी करने का काम जो होती है वो सेल वॉल ही करती है. So these are the some important function of the cells. It is outermost, protective, rigid, and it provides rigidity, and it also provides strength. Strength also given by this cell wall. And this cell wall, if we talk about the discovery of this cell wall, the Robert Hooke, the scientist who discovered the cell, he discovered the credit for the discovery of the cell wall is also given to Robert Hooke. So we can say that Robert Hooke discovered the cell in the 1665. This is the year when Robert Hooke first time saw the cell. He saw the cells in the Koch's cell. उसने Koch cell देखी थी जो कि ट्रिकअप बाग की cells थी. He saw some kind of compartments. इस तरीके के compartments उन्हें नजर आए थे, and these compartments have this outer part. This is the cell wall. उन्हें इन compartments से जो structure नजर आई थी, this structure is the cell wall. So the credit of the discovery of the cell wall is given to Robert Hooke. He called the compartment as the cell, and at that time the cell was these cells were the dead cells. Only the part that he saw is the cell wall. So Robert Hooke discovered the cell, and if we talk about the presence of the cell wall, where the cell wall is present, or we can say in which type of organism, in which type of cells the cell wall is present. So we can say that cell wall is found in the case of cell, we can say cell wall is found in the prokaryotes. In prokaryotes, cell wall is present. Here, exception is there. We know about the exception, that is the mycoplasma. Except mycoplasma, all prokaryotes contain cell wall, and cell wall is present in fungi, and in the case of algae also. In the case of eukaryotic cells, we can say that in eukaryotic cells only plant cells contain cell wall. So in the plant cells have cell wall, and some protists also contain cell wall. So some protists also can have cell wall as their outer membrane. So this is about some features, some functions of the cell wall, then discovery, and then we talk about where the cell wall is found. Now let's discuss about the composition of the cell wall, how the cell wall is formed in different organisms. Cell wall actually में बनी किस तरीके से है? We start from the prokaryotes. If we talk about the prokaryotes, in the case of prokaryotes, the cell wall is made up of peptidoglycan that we already discussed when we discussed about the bacterial structure. At that time, I told you that cell wall is made up of peptidoglycan. So we can say in the case of prokaryotes, cell wall is made up of peptidoglycan. And if we talk about the fungus or fungi, in the case of fungi, the cell wall is made up of chitin. यहाँ पे जो सेल वॉल बनी होती है फंगस के अंदर, वो बनी होती है काइटिन की, and this chitin is homopolysaccharide. It's homopolysaccharide of homopolysaccharide of N-acetyl glucosamine. In the case of fungus, the cell wall is made up of chitin, and chitin is homopolysaccharide of N-acetyl glucosamine. इसके homopolysaccharides means many N-acetyl glucosamine molecules join together to form the chitin, by which the cell wall of the fungus are made up of. In the case of algae, if we talk about the algae, in the case of algae, the cell wall is made up of cellulose. Cellulose is there, and galactans, mannese, galactan, mannese, and calcium carbonate is also present. So we can say calcium carbonate. These are the some composition of the algal cell wall. These are the substances which are present in the cell wall of algae. And in the case of eukaryotic, now we will discuss about the eukaryotic cell wall. In the case of eukaryotics, where the only plants have the cell wall is made up of cellulose. So we are talking about the plant cell. In this cell, plant cell is made up of cellulose, hemicellulose, hemicellulose. It has pectin also, it has water also, proteins and lipids. So this is the composition of the cell wall in the case of plant cells. In the case of plant cells, cell wall is made up of cellulose, hemicellulose, pectin, water, proteins and lipids also. This is the composition of the plant cells cell wall. Here, the cellulose is the homopolysaccharide of, it's the homopolysaccharide of D-glucose. It's the homopolysaccharide of D-glucose. D means dextrorotatory. So it's dextrorotatory glucose. Many glucose molecules join together to form the cellulose molecule. That's why we are saying here that it's a homopolysaccharide of D-glucose. The hemicellulose. Hemicellulose is heteropolysaccharide. It's heteropolysaccharide means it do not contain any single substance. एक ही तरीके का कोई substance join नहीं हो रहा formation में. It's heteropolysaccharide of different substances, like it contains galactose, it contains xylose, it contains mannose and arbinose. So this is about hemicellulose. Hemicellulose is heteropolysaccharide of galactose, xylose, mannose and arbinose. Here the pectin. This pectin is also heteropolysaccharide of glucosamine, glucosamic acid and glucuronic acid. Glucuronic acid. This is about the pectin. Pectin is the heteropolysaccharide of glucosamine acid and glucuronic acid. So this is the composition of the pectin. Besides this, water, proteins and lipids are also present in the formation of cell wall. So we can say in the case of plant cell wall contains cellulose, hemicellulose, pectins, water, proteins and lipids. Here cellulose is the homopolysaccharide of D-glucose, but hemicellulose is the heteropolysaccharides and it contains galactose, xylose, mannose and arbinose. Here now we will discuss how the formation here, that how the glucose molecules arrange together to form the cell structure of the cell wall. This is very important part. In the formation of cell wall, the glucose, these D-glucose molecules join together. Here almost 6,000 D-glucose molecules join together and they form one cellulose molecule. glucose molecules join together to form one molecule of cellulose. जब डी ग्लूकोज मॉलिक्यूल जिनसे सेलुलोज फॉर्म हो रहा है, इट्स द होमोपोलिसैकेराइड ऑफ डी ग्लूकोज. सो फॉर द फॉर्मेशन ऑफ सेलुलोज, डी ग्लूकोज मॉलिक्यूल्स हैव टू जॉइन विद ईच अदर. जब इस तरीके के 6000 डी ग्लूकोज मॉलिक्यूल्स आपस में जॉइन होते हैं, देन दे फॉर्म अ वन मॉलिक्यूल ऑफ सेलुलोज. दिस वन मॉलिक्यूल ऑफ सेलुलोज इज नाउ फॉर्म अ मिसल्स. ये जॉइन इस तरीके के सेलुलोज के मॉलिक्यूल्स आपस में जॉइन होंगे एंड दे विल फॉर्म द micelles. They will form micelles, and many micelles join together to form microfibrils. Microfibrils, and these microfibrils form later on the macrofibrils. And this is the macrofibrils. ये जो macrofibrils होते हैं, these macrofibrils are found in the matrix of the cell wall, which is formed of the hemicellulose. In the hemicellulose matrix, these macrofibrils are present. Here we can say that how cellulose is forming the cell wall. Cellulose is present in the form of macrofibril in the matrix of hemicellulose, and the arrangement of these macrofibrils in the formation of cell wall is different according to different organisms. किस तरीके से अरेंज होंगे वो सेल की नेचर पे डिपेंड करेगा. How these macrofibrils arrange to formation of this plant cell. So this is the arrangement how cellulose is joined to form the cell wall. We just say that cellulose is used for the formation of cell wall. हमने just इतना बोला कि cellulose से cell wall बनती है, but this is the complete way how cellulose form the cell wall. First of all, many D-glucose molecules join to form one cellulose molecules. Here 6000 D-glucose is joining to form a cellulose. Then cellulose joins to form the micelles, and these micelles form the microfibrils, and then microfibrils form the macrofibril. In this way we can say that cell wall is formed by the cellulose. इस तरीके से cellulose से cell wall की formation हो रही है. This is about the arrangement of the cellulose, how cellulose is forming cell wall in the case of plant cell. So here we discussed about the some important function, then discovery and the composition of cell wall in different organisms. Now let's see one more important thing here. In the some cases when these out of these substances, like हमने यहाँ पे देखा कि सेल बनाने में सेल वॉल फॉर्मेशन में सेलुलोज, हेमिसेलुलोज, पैक्टिन, वाटर, प्रोटीन, लिपिड्स, इन सब की रिक्वायरमेंट है. ये सब चीजें आपस में ज्वाइन होकर सेल वॉल की फॉर्मेशन कर रहे हैं. बट एट सम केसेस some other substances also get deposited on in the formation of cell wall. कुछ और substances भी use होते हैं cell wall की formation में. वो कौन-कौन से substances हैं? Let's see. वो कौन-कौन से substances जो cell wall में deposition का काम कर रहे हैं? They could be, यहाँ पे cutin हो सकता है. The other substances which are deposited in the formation of cell wall, that is cutin, and it could be suberin, it could be lignin. These are the some extra substances which get deposited with these substances for the formation of cell wall. When these substances are deposited, जब भी ये substances cell wall formation में deposit होंगे, then they provide some extra characteristics, or it depends on their condition which substances get deposited. Like if we take the example of this suberin. This suberin is the substance which is impermeable for water and the gaseous exchange. ये suberin जो होता है, ये जब cell wall formation में use होगा, जब ये cells में deposit होता है, then it makes the cell impermeable for the water. सेल को वाटर और रेस्पिरेटरी गैसेस के लिए impermeable बना देगा. जब सेल को वाटर और रेस्पिरेटरी गैसेस नहीं मिलेंगी, then cell will become die or it will become a dead cell. In the case of the Robert Hooke case, Robert Hooke ने जब सेल देखी थी, उन्होंने जो सेल डिस्कवर की थी, जो सेल्स उन्होंने देखी थी, that was the cork cells which taken from the bark of the tree. जो ट्री की bark से उन्होंने सेल्स ली थी. In these cells also suberin is also deposited. इन सेल्स में भी suberin deposited था. That's why these cells were dead cell. इसलिए तो हम कहते हैं कि ये जो cells थी dead cells थी. So these are the some extra substances which get deposited for the formation of cell wall. Here, let's see how the cell wall is formed. Cell wall is not a single layer. It is made up of three parts. Three parts combinedly form the cell wall. The first part is middle lamina, then primary cell wall, and then secondary cell wall. These combine, in some cases, tertiary cell wall is also formed. They arrange in a particular manner, and then they form the cell wall. Cell wall formation starts during the time of cytokinesis. If you remember the cell division process, जब cell division होता है, one cell divide into two daughter cells, and these two daughter cells get divided. First of all karyokinesis take place and then cytokinesis take place. जब साइटोकाइनेसिस होता है, साइटोकाइनेसिस के टाइम पे से दो सेल्स, एक जो सेल होती है वो साइज में बड़ी होगी और डिवाइड द ऑल द कंपोनेंट ऑल द साइटोप्लाज्म, एंड देन अ प्लेट अ सेंटर एट द मिडल ऑफ दीस सेल्स अ प्लेट लाइक स्ट्रक्चर इज फॉर्म, दैट इज मिडल लैमेला. एक मिडल लैमेला बनती है, वो मिडल लैमेला जो है वो बनी होती है फ्रेगमोप्लास्ट की. द फ्रेगमोप्लास्ट इज द सब्सटेंसेस व्हिच इज secreted by ER and Golgi apparatus, and this phlegmoplast is responsible for the formation of middle lamella. Later on, this middle lamella will form at the both sides of the middle lamella. Primary cell wall and secondary cell wall is formed. We will discuss about the complete formation, complete nature of this cell wall, how it makes in the next video. So keep studying, keep watching, and keep learning. Thank you.